Hi, thank you for choosing Videos.org. We're speaking to Sean Gibbons today about his toilet ecology. So, Sean, what can you tell us about your work?
Well, hi, thanks for having me. So I guess I'll start off by describing the field. It's sort of an intersection of two pre-existing fields. One is building science, been around for a while, studying how air currents and things move through buildings, how to design buildings. And the other is microbial ecology, which has really only got its legs underneath it in the last 10 years or so with the advent of high-throughput sequencing technology. So with these two different fields combined, we're able to explore and understand the dynamics of microbial communities that inhabit the spaces that we build, our built environment. And humans today spend about 90% of their lives in this environment. And we don't really understand exactly how that influences this new organ that we're now recognizing as a part of our bodies, which is the microbiome, this cloud of microbes that is in and on us that provides many essential functions like digesting our food and producing vitamins and so on. So we were really interested in how the interaction between humans and the surfaces around them fed back upon human health. So the study itself was in San Diego. It was two public restrooms, actually two male and female restrooms on two different floors in one of the student buildings there. And the idea was we wanted to know whether or not there was a reproducible ecological succession on these different restroom surfaces, and where are these microbes coming from? Do they form a stable long-term community that persists through time? With those basic questions in mind, the experimental design was to go in and sort of scorch the earth. We wanted to just sterilize everything on these surfaces. And to do that, we used a 20-minute bleaching process. We used microscopy to verify that there were no living cells left on the surfaces. And then once the surfaces had been scoured and cleaned enough, we allowed humans back into the restrooms, the dispersal agents. And so humans would use the restrooms, and then every hour or so, we would go in and sample a section of tile on the floor, the seat of the toilet, the soap dispensers, and the sink to see what types of microbial communities had developed there. So interestingly, what happened at first is a lot of fecal bacteria showed up. So right after cleaning, you get a huge pulse in organisms associated with our gut, which is probably due to aerosolization by the toilets. Every time you flush the toilet, a small amount of mist comes up, and that sort of disperses these fecal microbiota. So this transient pulse in fecal taxa happened, but then over time, they were displaced by the hardier aerobic skin-associated microbes on human beings. So the surfaces started to look like human skin, essentially. And we could even track the demographics of different restrooms, like whether males or females were using it, whether they were high-use or low-use restrooms, by the types of organisms that showed up there. So we could sort of infer human behavior or the types of people that were using the restroom by the communities that were there. And this feeds into a lot of basic research on this topic that has come out in the last few years, showing that human beings carry with them this cloud or aura of microbes that we are constantly shedding and dispersing into our environment. And we are able to detect those signatures of individual humans as they move through space. So we can see who was in a room, we can see maybe where they came from, and there's a lot of information encoded in this microbial cloud.
Okay, sounds brilliant. I mean, obviously my first question is going to be, what are the health and safety implications of this for the general public? I mean, is there anything we need to be aware of? I don't know, what do we need to do?
Yeah, that's a great question. And that was part of the impetus for the study. Can we design built environment spaces that maximize human health? And part of that was, is there even such a thing as a healthy built environment microbiome? What is the interaction between the surfaces and the humans? We know that humans deposit microbes onto surfaces. We don't know so much about whether or not they take them back up again, whether those microbes remain viable on these surfaces. So part of our study was to look at viability. So we had a section of the study where we excluded, after humans had used the restroom for a few hours, we then excluded human beings from the restrooms and sampled through time, and then cultured from those samplings. So culturing just means growing the microbes. So we wanted to make sure that microbes, not only were we detecting the DNA of these organisms on these surfaces, but these organisms were actually still viable and living over time after the humans had gone away. And indeed, we found that many of these organisms are still alive many hours after their dispersal agents are gone. So I guess the basic idea is that, you know, these surfaces are inhabited by organisms that normally inhabit the human body. Whether there's health implications there or not is difficult to say, because most of the organisms in and on our bodies are not only benign, but very beneficial. Many of them are actually needed for our health and well-being. Only a small fraction of organisms are pathogenic or bad for us. So I guess most of the organisms on these surfaces are our friends. For the subset that are pathogens, right, the microbiome of these surfaces reflects the health of the humans using these spaces. So if really a lot of sick people are using the space, they are dispersing potential pathogens, and those pathogens may remain viable. I would say we shouldn't panic. These surfaces are, for the most part, very benign, very safe, but still use proper hygiene. Wash your hands, because in public spaces, people are mixing their microbiomes, and if you don't want to mix with somebody who has an unhealthy microbiome, might as well just wash those hands. Just don't use antibacterial soap, because you want to keep your healthy indigenous microbes. They protect you.
Okay, so following on from that question, could this affect toilet designs in the future? Is this another implication of your work?
Building design or toilet design? Sorry. Toilet design?
Toilet design and building design of toilet spaces.
Sure, that's a great question. I'm not exactly sure. I don't know how you get around the aerosolization issue. It's a sort of small amount of mist that kind of comes up whenever you flush. It's not really a danger to our health, so I don't think there's a need to address it. But I think that toilets one day will be redesigned to be able to assess and monitor our health state through our microbiome. This is work that's being done by my current advisor, Eric Alm at MIT. They're trying to design smart toilets that will sample your stool microbiome every day as you use the toilet, and kind of monitor whether or not you're sick or healthy, or whether or not you need to eat more fiber, or you need to eat more greens, or how you should modify your diet to maximize your health.
So you mentioned the fact that everyone has the unique biome. Could this have forensic implications?
Very definitely it can, yes. So one of the recent papers that my group just put out, Simon Lax, a grad student in Chicago, was the lead author on this. Simon and I, walked around Chicago and sampled our phones and our shoes every day for about 48 hours. I guess it was like every other hour we sampled ourselves. And we wanted to really understand whether or not we could track where we had been and who was actually using a particular device or touching a particular surface. And we were very easily able to distinguish between Simon and myself. So his phone looked like his skin, which was a very unique signature and differentiable from my signature on my phone. So we could tell who was using what. We could also kind of tell who was walking where. If you were walking on the grass, if you were in a bar, if you were on the street, there were unique microbial signatures associated with the surfaces upon which we were walking that were reflected in the soles of our shoes. And there are actually several other studies, recent studies, that have done similar things with larger numbers of people that have been able to, with a fairly high degree of certainty, distinguish between individual humans based on their microbiome. So we are sort of microbial snowflakes, and we can use that to our advantage for forensics, perhaps tracking down criminals. If a burglar comes into your house and you can swab the kitchen counter where they put their hand and you can see their signature, you can prove that they were there. So I think this type of stuff will start to work its way into criminal investigations in the future.
That's brilliant. So what are your future research goals? What you got planned for the future?
So I'm kind of a generalist. I'm just kind of generally interested in microbial communities, their form and their function, how they change through time and space. I'm a theoretical ecologist who works in microbial ecology. So my future work, actually currently, I've narrowed my focus a bit to the human microbiome. So I'm trying to understand the dynamics of the human gut microbiome and whether or not we can learn in the changing of these communities through time, whether or not we can learn something about the health state of human beings and how to intervene and prevent or cure disease.
Okay, thank you, Sean. Thank you for your time today. It's been brilliant to talk to you. Your work's fascinating. To everyone out there who's watching this video, you can find the links to Sean's social media and the paper beneath this video. Thank you for your time. You've been watching ScienceVideos.org.